So today I'm going to talk about data-driven disease progression modelling. So this is modelling at a biomarker level to help us understand the progression of different diseases and for use for fine-grained staging and stratification. So first I'm just going to say a bit about big data in medicine. So there are now a large number of observational studies across diseases with a wealth of information being collected both in research and in healthcare settings. And these observational studies can provide us opportunities to gain new understanding of disease, as well as to perform personalised healthcare and to enable more effective clinical trials through identification of pre-symptomatic individuals for pre-symptomatic clinical trials. So the challenge with these data sets is to effectively integrate all the different types of information to learn disease signatures across a variety of scales. So here I've got some examples. So, wrong one. So you might have genetic information, also information from blood samples, demographic information such as gender, and different types of imaging measures. However, these medical data sets are actually quite challenging to analyse, so there's several difficulties in analysing this medical data. So one key issue is that often the disease groups are not well defined or they may be unknown. So you might have kind of very general labels such as Alzheimer's disease or in lung diseases, COPD, but these might actually constitute a range of different conditions that are underlying or different subtypes of that particular disease. And in the early stages, it can be difficult to identify individuals who are going to go on and progress to getting that particular disease. And these how individuals look at the pre-symptomatic stages may not be well understood. Another key issue is that the time axis is often not obvious or imprecise. So we can think about using clinical diagnoses, for example, but the clinical groups can be quite coarse. So I'm going to focus in this talk a lot on Alzheimer's disease, but I think these ideas generalize across a range of conditions. So in Alzheimer's disease, you would have, say, 3 diagnoses, cognitively normal, mild cognitive impairment, and Alzheimer's disease. And if you try and look at biomarkers across these different groups, you get a very low temporal resolution, so you've only got 3 stages in your progression modelling. Equally, if you try and measure against biomarkers, say a cognitive test score, no single biomarker would be sensitive to the disease stage across the full time course. So cognitive test score would necessarily not be sensitive to the pre-symptomatic stages. We also have issues with complex associations between different types of measurements. So these biomarkers are at a range of scales and they have vastly different complexity. So genetic and brain imaging information is very high dimensional, but then you can have regional measures which are much lower dimensional, and then blood measurements, which could just be single dimensional. So how do we associate these different measurements at different scales is another key problem. So in recent years, there's been a lot of development of machine learning tools and they're showing exciting applications. But these ideas often don't translate to medical data sets where the situation is just a lot more complex. So here are some examples of some classical machine learning or statistical tools. So the first is just classification. So here you might have 2 different groups and you want to learn a model that can separate these 2 groups. Actually, this has limited utility in medical data because you have to define the groups beforehand. So it necessarily requires knowledge of what the different groups are. Here's another example, which is regression. So here you have some kind of time axis, and then you're regressing biomarker values against that time axis to learn what the biomarker value is at different disease stages. However, this requires knowledge of what the time axis is and for the time axis to be quite accurate. Supervised techniques are more interesting, and for example, clustering, so just grouping data according to that data that has similar biomarker values. But these type of techniques don't fully exploit the structure of the data. So For example, they won't allow for temporal changes across the disease time course. They'll just associate individuals with similar values. So the aim of data-driven disease progression modeling is to develop new machine learning techniques that can exploit the structure of medical data sets. And these approaches have typically been phenomenological, so at a biomarker level, but incorporating the different scales is definitely important. So I'm just briefly going to explain the problem that disease progression modeling tries to approach. So here's an example in Alzheimer's disease of biomarker progression curves. So here we have a hypothetical model of Alzheimer's disease progression, and what we can see is that people think that going from the early disease stages to the late disease stages, you have different biomarkers that are sensitive to different stages of the disease progression. So they think that the disease starts with amyloid plaque build-up, then neurofibrillary tangles, and then in the later stages, changes in brain structure, and finally memory and loss of clinical function. So two of the common features of these types of models are a distinct ordering and severity of biomarker decline. And so disease progression modeling answers the question, can we learn quantitative models that describe these kind of ordering and severity of biomarker decline directly from the data? So why is quantifying these types of biomarker trajectories important? So there's several reasons for this. So the first is stratification for drug trials. So here I've got an example of a single disease, and the disease stage changes from, increases from left to right, and this is some kind of abstract measure of biomarker abnormality. So what we can see here is that the orange, blue, and red markers are early markers of disease progression, whereas the green marker doesn't change until a later stage. So using this type of model, we could perform stratification for drug trials by being able to identify individuals at a particular stage of the disease by comparing their biomarker values to this model. So we would compare their biomarker values and read off their most probable disease stage. We could also perform diagnosis and prognosis simply by, again, comparing biomarker values and reading off their disease stage. And we can understand the progression of the disease by identifying biomarkers that have shared so that vary together in time, so biomarkers that are closely linked, basically. So here's just an example of how you would perform that type of staging or diagnosis. So here, an individual with a high value of the orange and green marker, but a low value of the blue and the red marker, would match better with the kind of middle stages of the disease, but doesn't match particularly well with any disease stage. If we have a second disease, we can also compare to multiple diseases. So actually what we can see now is that this individual matches much better with the second disease, in particular the late stages of the second disease. So the most common statistical or machine learning approach that's been applied to this is regression. And this helps us recover quantitative biomarker trajectories by allowing us to plot disease stage on the x-axis, and then on the y-axis we have biomarker values, and we can simply extract progression patterns by comparing. So here's an example in familial Alzheimer's disease where people have used as the disease time axis time to onset of the disease. So this can be estimated in a familial disease by the age of onset of your parent. So in familial diseases, you've got a 50% chance of getting the disease if a parent has it. So you can perform this type of regression and extract curves for the different biomarkers. However, this age to symptom onset is imprecise. It's just based on your parent's age of onset. And this age of symptom onset can't be translated to sporadic diseases where we don't know when a person might develop symptoms. So here are some of the key challenges of reconstructing quantitative models. So the first is that often we have to reconstruct from snapshots. So, for example, in Alzheimer's disease, the progression occurs over, say, 20 years, decades. So often in observational studies, we only have a few years of data, so our observations are short with respect to the full disease time course. There can also be a great range of heterogeneity in disease stage. So like I mentioned previously, individuals within the same diagnostic group can be at a range of stages. So cognitively normal includes individuals who are pre-symptomatic, but also just healthy individuals. And MCI, some individuals may progress or some individuals may not. There's also a heterogeneity in disease subtypes, so individuals might have different phenotypes. So the idea of disease progression modelling is to ask the question, can we reconstruct the disease time course without requiring an accurate time axis that's derived a priori? So there are two kind of forms of these types of models, broadly speaking. The first one is discrete models, which work for purely cross-sectional data. And they model disease progression, for example, as a series of events where each event is a new biomarker becoming abnormal. And there's also continuous trajectory models, which reconstruct the kind of data-driven time axis as well as the curves of the biomarkers. And these require short-term follow-up in order to estimate the rate of decline. So the key thing about these disease progression models is that they're data-driven, so they don't require prior knowledge of disease stage, meaning that they're not limited to course clinical stages. Instead, they learn the stages of the disease directly from the data. And these models are actually applicable to a range of diseases as well. So here are some examples in multiple sclerosis, Huntington's disease, dominantly inherited Alzheimer's disease, posterior cortical atrophy, and sporadic Alzheimer's disease, and has also been applied to lung diseases as well. So first I'm going to talk about the simplest discrete model, which is the event-based model. And the event-based model asks the question, in what order do different disease biomarkers become abnormal? So it describes disease progression as a series of events. So each event corresponds to a new biomarker becoming abnormal. And this also derives a natural patient staging system where at each stage, the biomarkers up to that stage have occurred. So at stage 1, amyloid PET has become abnormal, and at stage 2, FDG PET and amyloid PET have become abnormal, for example. So how can we infer these kind of temporal progression patterns from purely cross-sectional data? So here's a kind of intuitive example. So say we have just two biomarkers, a yellow marker and an orange marker, and we can see that the yellow marker becomes abnormal before the orange marker. And now if we imagine that we're taking kind of cross-sectional snapshots of individuals that are at some stage along this model, if we take a sample at the beginning of the progression, we'll see that they have none of the biomarkers. Both markers are normal. At the end of the progression, both markers are abnormal. So neither of these tell us anything about the progression of the disease, but if we have samples in the middle of the progression, we'll see that the yellow marker has become abnormal, but the orange marker is still normal. So we can use this idea to infer that the yellow marker becomes abnormal before the orange marker, purely from just observing cross-sectional measurements. So here, if we observed cross-sectional measurements with yellow abnormal but orange normal, we know that yellow becomes abnormal before orange. And if we observe the opposite, so individuals with orange marker abnormal but yellow normal, then we know that orange becomes abnormal before yellow. So we can actually encode this idea mathematically to make a fully kind of probabilistic estimation of this ordering of events. So there's a lot of maths here, but I'm going to talk through it step by step. So the idea is if we consider that an individual is at a particular stage along a particular sequence of progression, so say they're at stage 1 and amyloid PET is normal, but the other markers are abnormal. Then what we can do for this particular sequence and stage is we can evaluate the probability that the events up to that stage have occurred and the probability that the events after that stage have yet to occur. So these just describe the probability of a biomarker being abnormal and a probability of a biomarker being normal. Then we can assume that this is a hidden disease stage, and we can describe the probability of the data given a particular sequence by integrating over this hidden disease stage. And then, assuming that patients' measurements are independent, we can derive the probability of the whole data set given a particular sequence. So now given this probability of a data set, given a sequence, we can try out lots of different sequences and derive what is the most probable sequence in which biomarkers become abnormal. So to do this, it depends on defining biomarker abnormality. So we can do this through a kind of data-driven mixture modeling technique, and from this we can get the probability that a measurement is normal and a probability that a measurement is abnormal, depending on an individual's biomarker values. So we then determine the sequence of biomarker abnormality by trying out different sequences and seeing which sequence best matches the observed data. So we have an equation for the probability of the data given a particular sequence, and we can then use MCMC sampling to determine what is the probability of a particular sequence given the data, and also the distribution of possible different sequences. So that gives us the uncertainty in the most probable sequence of events. We can then perform patient staging using this model simply by taking the most probable sequence of events and then evaluating an individual's probability of belonging to a particular stage along this model. So here, an idealized interpretation of this would be that at stage 2, amyloid PET and FDG PET have become abnormal, but hippocampal volume and MMSE score, say, have yet to become abnormal. So I'm just going to show some results from applying this model to Alzheimer's disease. So here we've looked at ApoE4 positive individuals who have a much higher likelihood of getting Alzheimer's disease. So this defines a kind of more homogeneous population group that better fits the assumptions that there's a single ordering of biomarker abnormality in the whole population. And we're looking at a general set of markers, including CSF measures, rates of brain atrophy, regional atrophy, cognitive test scores, and then just cross-sectional volumetric markers. So these are positional variance diagrams. So here, each entry in the diagram tells you the probability that a particular event occurs at a particular position in the sequence. If you have an entirely black diagonal, you have high certainty in the ordering of events, but grey blocks that change around have low uncertainty. So here this has high certainty that amyloid appears first in the sequence, but the hippocampus and entorhinal cortex have some probability of interchanging. We can also perform cross-validation of this sequence by bootstrap pre-sampling of the data and then refitting the model. And this gives us an estimate of the uncertainty in the sequence due to the variations in the data set. So what we find in Alzheimer's disease is that the ordering in ApoE4 positive subjects agrees with these kind of hypothetical models of Alzheimer's disease. So we see CSF measures becoming abnormal first, then rates of atrophy, then cognitive test scores, and then regional volumes. And we also see that so next we looked at the patient staging system to see whether it had utility for diagnosis and prognosis. And what we find is that staging individuals using this type of model, we get good separation of cognitively normal and Alzheimer's disease subjects. So cognitively normal subjects in green are generally assigned to earlier stages, whereas Alzheimer's disease subjects are generally assigned to the later stages, as you'd expect. And this gives a high classification accuracy for separation of those two groups. The MCI subjects, in comparison, are much more spread out across the stages and a much more heterogeneous group. We can also see that we can use this type of model to predict conversion between different diagnostic categories. So if we divide individuals according to their model stage at baseline and then look at their follow-up data to see whether they converted between different diagnostic categories, what we get is that increasing model stages correspond to an increased risk of conversion. So here, individuals that are assigned to stage 0 have a much lower probability of remaining MCI at follow-up, whereas individuals with high stages have a very high probability of converting to Alzheimer's disease. And the nice thing about this type of model is that it works across the full disease time course, so you can not only perform this type of prediction for MCI to Alzheimer's disease, but you can also perform it for cognitively normal to MCI. And so again, we can see that higher model stage corresponds to an increased risk of conversion. And this actually provides a good classification accuracy for both situations that's comparable to kind of machine learning techniques that are trained just on a single aspect of this problem. So machine learning techniques trained just for conversion from MCI to Alzheimer's disease. So now I'm just going to talk briefly about continuous trajectory models. So these models ask the question, what is the rate of biomarker decline, as opposed to the event-based model, which is discrete and doesn't quantify severity of biomarker changes. So one example of these is differential equation models. So these models work for just a single biomarker at a time. And basically they model, so you can assume that an individual's follow-up data is a differential cross-section of some average population trajectory. And then you can basically fit a curve to this differential samples. So you have the rate, the value at baseline and the slope, so the differential. And then you can integrate this to get the estimated trajectory of that biomarker for the whole population. And this can be formulated in stochastic ways as well to get an uncertainty in this biomarker curve. So probably the most highly cited example of this is this Villamagna's study in looking at amyloid PET, and what they show is that with this type of differential equation approach, that the time from being amyloid negative to amyloid positive is decades long. So that another type of model is self-modeling regression, which incorporates multiple different types of biomarkers simultaneously to estimate this kind of data-driven time axis. So here, if we plotted just age against biomarker values against age, what we'd see is that there's not really much coherence between the biomarkers and the age because individuals are at different stages. However, we can instead derive a progression score type of model, and by incorporating the fact that there's some time shift according to disease stage, we can estimate a much more kind of accurate biomarker progression pattern. So just a bit about how this works mathematically. So you have some kind of individual level model. So this tells you that your disease progression score is a function of the rate of decline of the individual and their time shift with respect to disease progression. So it's transformation of age according to how severe their disease is, basically, and the rate of decline. You then have a population model that's based on this disease progression score, which basically just describes the overall population trajectory, and then putting these two things together, you get an overall model that you can fit to a data set to simultaneously estimate individual speed of progression and population level trajectories. So here's an example. This is the first example of this type of model, which is the disease progression score model of Jedinak et al. And what they find is that RAVL-T is the first marker to become abnormal, and then amyloid and tau, and then cognitive test scores. This seems kind of surprising, and I think these kind of early models had a few problems with the fitting, but more recent models are not having this kind of problem of cognitive test scores becoming abnormal before CSF markers. So looking at the stages of the disease progression score, you can also see that you get good separation of different clinical categories. So in black, we've got cognitively normal, in red, MCI, and in green, Alzheimer's disease. And here's another example of this kind of self-modeling regression technique that's been applied to quite a range of biomarkers by Mike Donoghue. And this kind of resolves some of those problems that they were having with cognitive test scores becoming abnormal very early. I think one of the key things for these type of progression models is looking at more homogeneous populations. So here they just looked at ApoE4 positive individuals. So you don't have the problem of different individuals having, different underlying conditions, particularly in the MCI populations. So there's also spatiotemporal models of disease progression. So these ask the question, what is the spatial pattern of biomarker decline? So here's an example which is based on this kind of disease progression score idea, but it extends it for brain images. So this is work by Obergurgl et al., and it looks at a map of the rate of amyloid deposition. So here in each image you see the rate of amyloid deposition in particular voxel. And you can look at what this type of model would look like depending on different disease progression scores. So in the top are earliest stages, in the middle are sort of middle stages, and then the latest stages at the bottom. And this is kind of based on linear trajectories because the models are highly complex when you move to spatial models. So you can only estimate these kind of linear trends. Here's a nice example which extends this idea to have nonlinear trajectories. And the way that this is achieved is basically by finding clusters of trajectories that have common rate of decline. So here in blue, the voxels have the slowest rate of decline. In green, they have a kind of medium rate of decline. And then in red, they have the highest rate of decline. And grouping these together provides a kind of more compact representation of this disease progression model, which allows you to estimate these kind of nonlinear trajectories. So next I'm going to talk about disease subtyping, in particular some of the work that I've been doing on subtype and stage inference, which is asking the question, firstly, what are the different subtypes of a disease, but also simultaneously, how do these different subtypes evolve over time? So here's a kind of schematic of the underlying problem that we're trying to solve. So it's based on the assumption that there's some underlying disease process where there's two groups of individuals, or however many groups of individuals. Here there's two. And that these groups of individuals have different progression patterns over time. We then have a data set, which is basically a heterogeneous set of patient snapshots of this underlying disease progression pattern. So you have individuals from the different subtypes and from the different stages of each subtype. And the problem you're trying to solve is reconstructing what is this underlying disease process. So typical machine learning approaches that have been applied to this problem previously are firstly clustering, which just associates individuals with similar biomarker values. But you don't get a differentiation of what is a disease subtype and what is a disease stage. So here we can see that you might have the orange subtype, but at a range of stages in one cluster. Another cluster might correspond to a mixture of subtypes at a moderate stage, and then another cluster might correspond to another specific subtype at a range of stages. On the other hand, you have disease progression modeling techniques, which assume that there's a single progression pattern for all individuals, so they don't have subtypes. So the idea of subtype and stage inference is to integrate these two ideas to simultaneously estimate both subtypes and progression patterns. So basically, this is just a stratified model of disease progression. So rather than having a single disease progression pattern, we now have multiple disease progression patterns that we're trying to estimate from the data. And the specific way that this is formulated is as a mixture of piecewise linear progression models. So we have a model where biomarkers transition between different z-scores. So you have a kind of notion of severity of decline. And it's basically a form of semi-supervised learning where you only require a control population. So what we do is we input the heterogeneous patient snapshots, and then we get an output of the reconstruction of disease subtypes and stages. So we do this by fitting increasing fixed numbers of these progression models, and then we can actually evaluate the optimal number of subtypes using cross-validation. Given this type of model, we can then apply it for subtyping and staging of new individuals. So brief example in Alzheimer's disease. So applying this model just to regional volumetric imaging in Alzheimer's disease, we get these three different subtypes. So here, each row is a different subtype, and then each of the, going from left to right, you have the different stages of that subtype. So the first subtype is a typical subtype, which has kind of both hippocampal and temporal lobe atrophy. Then there's a more cortical subtype that tends to affect cortical regions earlier in the progression, and a subcortical subtype that generally affects subcortical structures more severely. And these subtype patterns actually reflect nicely with neuropathological findings of these subtypes. So we have this kind of typical cortical and subcortical subtype, which correspond to specific stages of these subtypes correspond to the typical hippocampal sparing and limbic predominant subtypes that have been observed at autopsy. But the nice thing about this type of progression model is that you get the full temporal progression, so you can stage individuals as well as subtyping them. So we had a look at the utility of the subtypes and stages for basically diagnosis and predicting conversion. And what we find is that the sustained subtypes become more, so subtype and stage inference sustain, become more identifiable as the disease progresses. So in cognitively normal individuals, you can see that, so these are triangle plots, and in the middle you have an equal probability of belonging to a particular subtype, and then at the corners, individuals are strongly assigned to a specific subtype. So cognitively normal individuals are generally in the middle, meaning that they can't be identified as a particular subtype. In the MCI subjects, we've got some that you can't identify, but equally there's some clusters of individuals starting to appear that are strongly assigned to a particular subtype. And then in the Alzheimer's disease subjects, you get a strong assignment to specific subtypes. And these, both the subtypes and the stages of the model have added utility for patient stratification. So again, looking at your subtype and stage at baseline, you can use this to estimate the probability of converting from MCI to Alzheimer's disease from follow-up data. And what we see is that your subtype influences your probability of remaining MCI. So the typical subtype are most likely to progress from MCI to Alzheimer's disease, and the subcortical subtype are the least likely. And also your stage influences your probability of remaining MCI, as we'd seen with previous models. So the final thing I'm going to talk about is network models. So these are kind of at the level of brain networks estimated in healthy controls and looking at how this disease propagates along these networks. So they ask the question, how do diseases progress along different brain networks? So broadly speaking, there are two approaches that network models have taken. So the first is testing different mechanistic hypotheses. And the second is looking at a specific hypothesis and then simulating the propagation and comparing the propagation patterns that are predicted to the actual observed propagation patterns. So here's the first example of testing different mechanisms by Zhou et al. In 2012. So what they do is they derive a healthy connectivity graph and then basically look at the network vulnerability based on different models and how this relates to actual atrophy that's observed in the disease. So they have several different hypotheses. So the first is nodal stress. So they predict that if your disease progression is influenced by nodal stress, you'd see an increase in graph theoretic measures of the total flow along the network. Whereas with if it's a transneuronal spread mechanism, you'd see a decrease in metrics showing the shortest path to the epicenters. Then for trophic failure, you have decrease of total flow and decrease of clustering coefficient. And for shared vulnerability, they don't identify any predictions. This is kind of schematic of how this type of study is constructed. So you have different atrophy maps for particular diseases. So here the example is behavioral variant, FTD. And then you're looking at connectivity in healthy subjects. And then looking at their graph theoretic measures and comparing this with the observed atrophy pattern. So they tested different mechanisms for a range of different types of conditions. So here they're looking at, so the top is the total flow measures. So for each node in the graph, you can look at the total flow and correlate it with the atrophy pattern in that region, in that voxel. And then, so the total flow was indicative of nodal stress. The shortest path to epicenter is indicative of the transneuronal spread hypothesis. And the clustering coefficient was the trophic failure. So what we can see here is that, you see, in all of the diseases, or the majority, most of them correspond to this shortest path to epicenter in health, suggesting that there's this kind of transneuronal spread pattern. But some of them have this kind of more total flow in health pattern, suggesting that there's just failure of nodes under nodal stress. So the kind of opposite approach is simulating progression along brain networks. So this model is based on the assumption that of the transneuronal spread hypothesis, so kind of prion-like progression. And it basically performs the same type of analysis, but you simulate what the progression pattern would be and then compare with the outputted atrophy patterns. And what they find is that when you simulate this kind of network diffusion model under the transneuronal spread hypothesis, that you can get different eigenmodes of this network diffusion, so kind of different components of the network diffusion model, and that they actually correspond nicely with the atrophy pattern measured in different types of conditions, so here in Alzheimer's disease and here in behavior variant FTD. And this is the same kind of picture as previously, where they're showing that this the atrophy predicted by a particular aspect of the diffusion model is best correlated with a specific disease. And these models, particularly the second type of model, have been extended a lot recently to model various different types of mechanisms, including amyloid clearance, for example, and build-up. So I'm just going to say a little bit about future challenges of these kind of models. This is by no means comprehensive, but just highlighting a few things. So the first one that I've picked is integration of different data types. So at the moment, progression models have mainly focused on this kind of phenomenological trends, so just looking at a biomarker level. But ultimately, to personalize these type of models, what we need is to combine these kind of time-dependent measures with also other kind of fixed variables. So, say, for example, genetics or demographic and lifestyle factors to model how these influence these progression patterns. This is actually quite a big challenge because things like genetics have very high dimensionality, so you need kind of approaches that can extract relevant features and make parsimonious models that share different variables and can identify a wide range of associations. So the next thing I put is propagating information from one scale to another. So, for example, by using network models to influence what your biomarker progression patterns might look like at a higher level. Or equally by using progression models to refine your network model. So you could potentially have much more complex mechanisms in your network models if you can compare to this kind of more fine-grained biomarker progression pattern rather than just late-stage final atrophy values. So another challenge is simultaneously learning new features in progression models. So this relates to this kind of personalization problem in terms of genetics, but also there might be new imaging features that you could learn. For example, extending these ideas to deep learning to automatically extract your imaging features and also the temporal progression. And then the final thing is sharing features across the models. So in comorbidities and in highly complex data, you want the most compact representation you can possibly have, and this would be achieved by sharing different variables. So another key issue is translating these types of models from the research setting to healthcare, and this is actually incredibly complicated, and there are several different problems that surround this. So one of them is extracting useful information for clinical practice. So highly complex models might not be very interpretable, and they provide, say, too much information for a person to really comprehend in a short clinical workflow. So ideally what you want is just key bits of information, and then maybe the opportunity to look in more depth at specific measures if there's some kind of confusion in the diagnosis. And this kind of leads directly into the idea of designing the best diagnostic aid, which is also incredibly tricky. So here's an example that someone made in Alzheimer's disease, where they have a kind of disease index, and then they have different probabilities of different biomarkers becoming abnormal. And then you can kind of click on different things and visualize what the different measures look like and how an individual's measurement compares to the population measurements. But actually choosing what is the relevant information and how to design the system is very difficult and requires close consultation with clinicians to see what information is most useful. Another key issue is harmonizing the different disease signatures that are learned in research with those in healthcare. So often there's quite different types of information available in healthcare, and or different types of scanners, for example. So how can we move the models from research to healthcare? And then final thing that I think is actually really interesting is designing systems that optimize biomarker utility. So currently disease progression models just look at, take a set of biomarkers and look at the trends in that set of biomarkers, but you can actually imagine a system where you could say, depending on the cost of a particular biomarker, you could go through and get your model to optimize what is the best set of biomarkers that I could take for the least money that would help me identify, say, a particular stage of the disease. And I think that's quite an interesting problem to consider. So yeah, thanks, and questions.